Hi everybody, welcome to the third talk of GA Day. I'm Larissa Hayes, I'm a PhD student here at UFG, and I'm really happy to be here with you today. It's been a very productive morning so far, right? And to wrap up the first part of this event, I'm delighted to welcome and present Dr. Daniela Bustos-Korts. She'll be presenting on the topic, deciphering G by E from KTL to prediction in real environments. Dr. Bustos-Korts is an assistant professor at the Austral University of Chile in Valdivia. She holds a master's degree in plant science and a PhD in statistical genetics. Before joining the Austral University, she worked as a postdoctoral researcher and assistant professor at Wageningen University and Research. Her research focuses on modeling genotype, environment, and management interactions and phenotype prediction in crops, especially wheat. Before we begin, I'd like to remind the audience that we'll have questions and answer sessions, and feel free to submit your question through this YouTube chat. Our team will read and select some answers so we can discuss them later. Daniela, it's truly a pleasure to have you with us today. Thank you so much for taking the time and sharing your expertise with us. You can start your presentation now, please.
Okay, thanks a lot, Larissa. Well, I hope everyone can hear me well. I have a bit of a sore throat, but I will do my best to make it as clear as possible. I will then share my screen. So here. It's coming. Can you see my slides? Can you see the slides?
Yes.
Yes.
Could you just close the meet box in?
This one?
Yeah, this one.
Yeah. Perfect. Thanks. Well, I took a bit of freedom to modify the title because I thought, well, it's nice to talk about QTLs, but we need to understand how these QTLs are embedded into something else. So I expanded it a little bit than it was originally meant. And well, the title of this talk will be From QTLs and Dynamic Traits to the Target Trait, Modeling G/E/M Interactions for Complex Traits. So, well, I was already nicely introduced, but as Larissa announced, I'm currently based in Valdivia in the south of Chile at Universidad Austral. This is a very green city, a university city. We are near the ocean, and so there is a lot of nice things to do regarding tourism and also some nice beers. And it also has one of the largest potentials for wheat production worldwide. So the yields here are very, very high. And this is why it's also very interesting to study how cultivars can adapt to this kind of environments. So let's enter to the topic. So perhaps, well, you already know you are all plant breeders, so this will be very familiar to you. But the goal of plant breeding is to select or to produce and select varieties that perform well in a target population of environments. So it's always nice to think about little trials, but we always need to think back and say, well, how do these trials that I am performing in the greenhouse, in the field, or in another place represent the growing conditions that are relevant for those varieties when they are going to be grown by farmers, so the target population of environments. So we have a distribution of soil, weather, management conditions, and the combination of those we try to represent by these multi-environment trial networks that we select a number of locations, years, and management conditions that are as similar as possible to the future growing conditions to increase genetic gain. So nowadays, in the past, it was a lot about phenotypic selection. Now it's a lot more about prediction-based. So we have these multi-environment trials that represent the growing conditions, the target population of environments, and the germplasm that is relevant that we have at our disposal as plant breeders, and we test them. But we don't want to make selections only on those genotypes, on those environments. I want to use those data to predict how new varieties would perform, or how a current variety would perform next year, for example. Yeah. So at the end, this means we are transforming, as proposed here by Cooper in these nice papers in 2023. We are moving from something that is based only on phenotypes, that the genetic gain is defined as the selection intensity times the heritability of the trait, how much variation we have, and time. Then we switch to something that is more related to prediction accuracy. So how precisely and how well we can predict the adaptation for another location that was not tested, or for another year that was not tested, for example. That's one thing. But then we also need to go a little bit beyond, because if I predict for, I don't know, Valdivia 2025, that's okay. But I also need to think, how does the trial that I performed to train those models represents or not the TPE? And this is why in the paper by Cooper, they add this component of correlation. So how similar are the testing conditions from those that are to be expected in when grown by farmers? And then we really have the full picture of what genetic gain could be. It's slightly more complex than what we were thinking a few years ago. But doing this is not very simple, because, well, this is a GBI symposium, so probably you already heard a lot and you know a lot, but still it's good to define what GBI is. So GBI is basically, we characterize how genotypes respond to an environmental gradient. And we could have situations, for example, in which we have no plasticity, so the trait does not change, and they don't have GBI, so everyone stays the same everywhere. This is usually for traits like flower color, things like that. Then we could have plasticity, so if we change the environment, if we improve the environmental condition, we add fertilizer, water, whatever, the genotypes improve their yield, but everyone improves in the same amount. So we have plasticity because there was a phenotypic change, but there is no GBI. So we always prefer the red genotype, or genotype 1 in this case. And we have a lot more complex situations. Of course, these are still quite simple because they are very linear, very nicely behaved, in which we have different sensitivities of the genotypes to the environment. And the most complex one, as you probably know, is when we have these rank changes. And the challenge is, how can I understand what is causing these rank changes? So is a certain groups of locations? Are there certain management conditions that modify or make that, for example, in low quality environments, the green is the best, and in high quality environments, the red is the best? Or we could be in a more complicated situation, and I will dedicate large part of my talk to describing techniques for this more complex situation in which they are non-linear. And then we really have a challenge, because it's very hard to understand for which set of conditions I would prefer one genotype or the other. Yeah. So a lot of this talk will be dedicated to techniques that, or different strategies that help us understanding how can I make groups of environments that help me selecting more effectively, so that I can increase this genetic gain within each of those groups of environments. So predicting GBI/M, there is a lot to do. It's quite a challenge, but I try to group it in two set of things. One thing is how we can understand the structure of this target population of environments. Do we have mega environments? Do we have water stress scenarios? Do we have trends in space, latitude, longitude, or time, for example? And the other part, and this more mechanistic perhaps, is the physiological and genetic drivers for genotype adaptation. So which are the traits or the mechanisms of response, and how these mechanisms of response are related to environmental conditions? Yeah? How can we model traits over time? And then finally, all these things, we try to get to some QTLs that drive these responses over time and trait correlations. Okay, so let's get started with the first part. So the first part, the logic here, I try to think about it is, well, we try to group trials into mega environment scenarios, but if these mega environments or groups of environments really have a particular type of stress, for example, well water and water stress, then the same traits should be driving adaptation to those environmental contrasts. Yeah. So in that way, looking at how different traits relate to each other can be helpful. For example, if you have late droughts, flowering time can be of particular interest. Why? Because genotypes that flower early might escape that late drought, whereas genotypes that flower late will encounter drought in the flowering season and then yield will be lower, for example. Yeah. But how to analyze this is not so simple. There are many techniques. One of them, well, linear mixed models. Maybe you are familiar, things like the factor analytic model or other type of very useful models. And here we show some examples, particularly of the self-organizing maps or other clustering techniques that could be used. This is just one out of many that you can have, and splines. Yeah. So maybe some of you are familiar with self-organizing maps already. So basically, we have some unsupervised learning algorithms. So think about something like a principal component analysis. In a principal component analysis, you can have 2, 3, 4, or many dimensions, and becomes a little bit difficult to visualize it. In a self-organizing map, it's more or less the same idea, but you sort of stretch the multidimensional plane in a grid. And you put it, this is why it's called a map, because it's in 2 dimensions. And what happens with this map? Basically, you assign your observations to units. And units are certain, we call them prototypes. So a certain pattern of response. For example, I could have trials in which all the traits have a low value, so low yielding trials. Or I could have trials that everyone has a high yielding value. Or I could have trials like this one in which all the traits have low value except great number. And in this way, by looking at the similarities between the different traits in set of trials, we can group them. Yeah. And the nice thing of this is, and why it's, I think, useful for plant breeding, because as breeders, we not only have quantitative traits like yield, biomass, days to flowering, we can also have other traits. For example, does it have a disease or not? So yes or no traits, binary traits, or we could have categorical traits, flower color, type of, I don't know, certain particular morphological trait, etc. And here in self-organizing maps, you can have different layers of information. You could have genotypic traits, and you could have other traits that have, for example, even environmental traits. And you have one matrix or one layer per type of information. And that is very nice, because then you can have a different similarity measure for each type of traits. For example, Euclidean distance for quantitative traits, or other kind of distance for other type of variables. Yeah. And then the overall distance is weighted average of the layer distances. So you combine the information for the genotypic traits, disease course, environmental variables, etc. We tried these ideas in a large data set for sunflower. And we basically, we said, well, this idea is very nice, but in real data, things are always more complex. And one of the first complexities we encountered is the fact that a different location is sampled every year. So the trial is not always exactly at the same location, but maybe a farm a few kilometers away. So we said, well, how to deal with this? Because as you maybe know, as G/M modelers, we need to have some overlap. We need to have some trial connectivity, that we have the same location tested at least a few times, so that we can really test how genotypes can respond to this, or how locations or years induce G/E variation. So the first thing we said, well, these locations actually represent more or less the same thing. So we clustered them based on latitude, longitude, and we said, well, all those that are in pink here represent a sample of a relevant production region. Yeah, and then we will look G/E and at these regions level. Yeah. So then we looked at these self-organizing maps, and we used each genotype. We said, well, actually, if we want to group locations, each of the genotypes is one probe, one way of testing how good that environment was. Yeah, and we have some contrast. For example, here we see that genotype 22. Here we have three traits: yield, oil concentration, and thermal time to flowering. We saw that in general, genotype 22 was a late flowering genotype. Why? Because in all the environments that are represented by the circles, it has a large radius here. Whereas genotype 16, for example, has a small radius here, which means it is very early genotype. But in contrast, genotype 22 has very, very low values for yield and oil, because it's reflected especially in these gray environments. We have low radius here, and in the green and blue environments, we have high values for yield, for example. So we see one first pattern: poorly adapted to the gray and yellow, well adapted to the blue. Genotype 16, in contrast, is different because it's well adapted to most of the environments. It has large values of this green part. So we did that using all the genotypes, and then we combined the information, and we drive to a cluster that says, basically, you have gray trials in which each genotype behave in a similar way, and you have the blue trials that you have other patterns of response in which other genotypes, for example, genotype 22, would benefit. Yeah. So, for example, in gray trials, you would prefer genotype 16 or genotype 8, and in blue trials, you would prefer genotype 22. But these are just trials. We are looking at combinations between locations and years. And we wanted to group locations. That is slightly different. So then we said, well, but if we get a different group every year, we can look at the long-term frequency. So in other words, how often this set of locations behave like gray or behave like blue. So then we clustered based on these frequencies of occurrence of a certain pattern. Here we are looking only at rankings. So in how often we would prefer this genotype in that location, how often we would prefer the other genotype in that location. However, if we would go in more detail, and if we would have environmental conditions, then we can try also to relate and see, well, actually, when we have, I don't know, this weather pattern, we would find the gray environments, and then we would prefer genotype 8. When we have the other weather pattern, for example, less rainfall, I don't know, yes, same, we would have the blue pattern, and then we would prefer genotype 22. So in that sense, we can try to relate environmental drivers to these clusters' frequencies. So this is what we did. So we looked at these frequencies and we said, well, actually, here in France, for example, they are very similar in the sense that these two sets of locations or trials have a predominant, or the proportion between gray, blue, and other type of environments are similar. So they are characterized as blue. In contrast, we have other environments in which the yellow is the most common, and then we form another group of locations, and actually this one and this one are quite similar because they have yellow and gray, so we put these two sets of regions together. Yeah. So then we go from something that has variation, that is genotype and year, because here we see, location and year, because here we are having a set of locations that vary across years. Some years they are purple, some years they are gray, some years they are blue, to something that is just regions. We say, okay, in this region we have a certain probability of this stress type to occur. Yeah, and that's very nice, but the big question is, does it work? Because we can always make nice groups and have a nice map, and we say, okay, we have this part in France that is different from the north of France, but does it help us to explain G/E? That was the big question. And how can we evaluate this? Well, then it's always good, once we have the groups based on the self-organizing maps or on another clustering technique, we can always go back to the mixed model. And the mixed model tells us how important that region is. Yeah. So here we put it back in a fixed part that has location, year, location. And then we have the genotype, the zones, and how the zones react also or vary over years. And then we can assess to which extent the heritability, when we calculated ignoring the regions, is different from the heritability that we have when we add the regions. So the heritability with these environmental zones. And then we can calculate the genetic correlation between the regions, and we can come up with this equation that was proposed by Adlin a few years ago already, many years ago already, I think it was almost 20 years ago, to check what do we gain in terms of response to selection when we select for specific adaptation to each of the regions, whereas when we select ignoring this specific adaptation to each of the regions. And we had high expectations, but reality is always a bit more difficult. In spite of this being a very nice data set, quite large, many trials, well-selected genotypes, it was hard to make groups that explained a lot of G/E. So here we checked what do we gain, and we said, well, actually, if we ignore the regions, we would have a prediction accuracy 0.42. And if we include it, we will have a slightly better accuracy. So it was better. So in most of the regions, we do gain from selecting specific adaptation because we get these values that are higher, but yeah, not as high as we wanted. But well, that's reality. Still, it seems it pays off to split this big mega environment network into subsets. Now, so that was one thing, to group locations. And then I will quickly show another technique, more looking at a continuous G/E, and this is based on splines. So what are splines? I will use it in different contexts in this talk. The first one is to group of environments. And splines are basically piecewise polynomials that are defined in a certain range. That could be linear, for example, here, quadratic, or cubic. And we have that these piecewise polynomials, they are covering the whole range. And then we have some coefficients that basically stretch or shrink these piecewise polynomials so that we can fit the data. Yeah. So here, for example, these points can be explained perfectly from the sum of this blue times a coefficient, and then the purple and the red. If we sum the contribution of these three piecewise polynomials for this value of x, we obtain this one. But if we do that very much in detail, we will overfit, and we don't want that. So this is why, well, the optimal penalty is embedded in a mixed model formulation, and then we have a smooth trend that enable us to characterize the overall response, but not so weakly, a bit more smooth. Yeah. So well, this is embedded in a nice R package. If you are interested, you can look at it. It's LMM solver that was created by my colleagues Martin Boer and Bardian van Rossum, and there are some papers also to it, connected to it. So what do? We say, well, if we go back to this idea of adaptation landscape, so basically looking at how a genotype will react to a certain environmental gradient, we can fit a spline. And here we have different genotypes in Australia. And here we have latitude and longitude for one genotype. And we see that this genotype, for example, is in general high yielding. So we have all blue, green and orange colors, and especially in this part that is more close to the coast, is especially high yielding. We have other genotypes that are always bad, for example, like this one, always low yield, no matter where. And we have others that are also intermediate, but we have some, particularly in the north, for example, and in the south, that are higher yielding. So if for each genotype we could know how it would react, so how yield would change if we move from north to south or from east to west, then we can put all these layers together for each genotype and check, okay, for this latitude, I would prefer that genotype. For this other latitude, I would prefer this genotype in the picture H, for example. Yeah. So if we do that, for each point in space, we can look at the genotypic ranking, and we can group locations based on that ranking. So we did that in two type of years. So we have the mild year, in which we have intermediate values of rainfall, and we did for hot and dry years. And we see that when we have hot and dry years, we have more subdivisions in our TPE, basically because when we have less water, then the soil conditions are exposed, and we can have more differences in the latitudinal and longitudinal gradient. Yeah. So these groups are based on basically which are the top 5 genotypes that are presented for each point in the map. Yeah, we see, for example, all these locations would share the same top 5 genotypes in a hot dry year, whereas in these parts, then they would share another top 5 genotypes. Yeah. So again, we need to go back to the linear mix model and check, does it work? And we saw that in general, well, for mild years, this cultivar by region was quite successful in explaining G/E. So basically, the contrast between the purple and the green was very good in terms of explaining genotypic ranking, whereas in hot and dry, this was not so successful. So this is also why it's important to consider the year type and to see if we have in Chile, for example, well, also in Australia, we have the Niño and Niña years that are very contrasting in their phenology. So it makes sense to make regions that are specific for Niño and Niña years. Yeah. So that's one thing. And then if we can zoom into the mechanisms, because until now we are only looking at a few traits, but we are not looking so much at the mechanisms, and we also didn't look at the QTLs yet, only phenotypes. Yeah. So here we need to keep in mind that when we look at traits, it's not every trait the same. We have a hierarchy. So we have yield that is quite complex, regulated by many QTLs, and also has a more polygenic basis. And then we have basic traits that are more the response mechanisms. For example, how much a cultivar responds to the day length. Yeah, probably a few genes only. So it's more predictable in the sense that it has a non-genetic basis and less GBI. Yeah. So one of the things that can help us understanding this hierarchy are these graphical models. So here I'm sharing some results from my former colleague William Kravier. There is also a nice R package attached to it if you want to look. But the basic idea here is we have many traits from genotype to the most complex one, that is yield, and we want to understand which one is causing which one. Yeah, and how this causality changes depending on whether we have dry environments or non-dry environments. Yeah. So in this case, we were playing around with some simulated data, but we could also do it with real data. And we have here these basic traits that are directly connected to QTLs: photoperiod sensitivity, radiation use efficiency, water uptake, etc. And then these complex traits that are created from these basic traits. And we see that the hierarchy is correctly identified by the model. Why? Because we have all the grays at the top. Those cause the green, and those cause the target trait that is yield. Yeah, and we see that the traits that are relevant for yield differ. And the dry conditions, we have the water uptake capacity of the plant that is driving biomass, and biomass is driving yield. And when we don't have drought, we have radiation use efficiency that drives biomass. And the water uptake capacity is not so relevant because there is always enough water, so it doesn't matter how efficient the genotype is. So that can help us prioritizing traits for each of the environments. Here I'm showing also some nice work from a former colleague. Her name is Yutaka Susumi. She was in Bethany and now she's back home in Japan. And she tried to integrate trait dissections to explain GBI. And she had quite a complex tomato cross using cultivars and wild genotypes. And then she produced some reels, so that were coming from these four parents, and they were tested in different environments. And then she was dissected. We have yield that can dissect it in the number of fruits, individual fresh weight, or we could have another physiological dissection in which we have total yield that is dissected into fruit fresh dry weight ratio and the total fruit dry weight. So how much fruit biomass there is, and that depends on the harvest index, so how much fruit versus leaf and stem, etc., and the total biomass. Yeah. And why do we want to do this? Well, because complex traits have a complex, obviously, physiological basis, and many times we have negative correlation or trade-off. For example, if we increase the number of fruits, it also happens that we get smaller fruits. if we increase, if we have a larger value for fruit fresh dry weight ratio, so we have also less total fruit weight. The same happens in other species. For example, I work in wheat, and then we see if we have more grains, the grains become smaller. So this is not exclusively to tomato, but in many species we find that we try to improve one trait and then the other one is reduced. And then we have a compensation and we end up with the same yield. And we don't want that. We want to have those that have high values for all the traits so that in total we can have high yield. That means going up in these lines that represent higher yields. For example, those individuals here in general have higher yields. So what did Yutaka do? She tried to find QTLs. But QTLs in here, remember, we had 4 parents. So actually we had 2 elite lines and 2 wild parents. So each of them, we hope, could have a certain positive contribution so that we can select pieces from here and there to make the perfect genotype that would have the highest yield possible. Yeah. So here we have, well, different chromosomes, and we see, for example, here that we have the 0 line. So all those alleles that are above means they are good. Yeah. They are good for yield, the marketable yield, so the nice tomatoes, the number of fruits, and the individual fruit fresh weight. Yeah, and then we can see that in most cases it is true that the lead lines have the positive alleles, so most of the alleles here are positive, which makes sense because breeding has worked a lot on those. But it's also the case that for some of them, the good alleles come from the wild cultivars, the wild accessions, so the wild parents. So that means that indeed making this complex cross can help us increasing the different traits. And if we can add, for example, if we select for the number of fruits, that can also help total yield if we reconstruct this hierarchy. So we can apply some selection pressure on the yield components or on the yield itself, and that would increase our genetic gain. So here we are dealing with some static dissections, and we are trying to connect those static dissections with QTLs, but I need to speed up a little bit, I think, to yield components. What we can also look at phenotypic data over time. And now we have a lot of techniques to do that with drones and other sensors, etc. But the challenge is how to summarize them, because we have a lot of pictures and a lot of pixels, but each of those pixels carries very limited amount of information, and the challenge is to remove the noise and to create summaries or parameters that are as predictive as possible. Yeah, so well, perhaps you are already familiar with the spatial adjustments, but one of the first things to do is, of course, to remove trends in the field or experimental factors that could mean noise, and we concentrate on the genotypic values. And once we have those genotypic values, we model them over time. So for example, here we have a small cartoon. We have genotype 1, that is the curve does not change a lot if we have them in 2 water conditions, whereas genotype 2 changes a lot. It's dynamic if we move from one water level to another one. In contrast, genotype 1 is very sensitive to temperature because it changes a lot. It's dynamic over time if we change the temperature, but genotype 2 doesn't matter. It stays more or less in the same dynamic regardless the temperature. So if we do that, after removing the experimental noise, and we extract those parameters, we can try to find QTLs for those parameters. Yeah. And here, well, my former colleague, Emilie Millet, she was working in a nice package that is called StatGen HTP. You can find it on CRAN to model how these dynamics can occur for the whole population or for each of the genotypes. And the idea is to integrate those curve parameters into something that is like a response surface, and at the end to recover the whole G by E landscape that combines the different traits and the responses of those genotypes to the environmental conditions. So this is a little bit what we tried to do some time ago, actually when I was still doing my PhD. We said, well, we have this thing that is very complex. We have a lot of G by E that depends on many traits, and those traits change to the environment. What to do? Let's simulate something. And we did a sample of the target population of genotypes, a sample of environments, and we have QTLs. And those QTLs were the same for all the environments. For example, photoperiod sensitivity was the same for all the environments. Radiation use efficiency, the same for all environments. But when we combine through the crop physiology model that parameter with how much radiation there was, how much temperature there was, etc., we create this complex target trait that was having a lot of G by E. Yeah. So then we said, well, we put QTLs that were constant over time and that were static across environments, constant across environments. What happens with the intermediate traits? What happens with biomass, for example? Here we see the correlation between the different, so this is more related, the phenotypes and grain yield. So for example, here we see to which extent biomass correlates to yield. We see that in this environment, we always get a positive correlation. This environment did not have any water stress. But if we move to an environment with a strong water stress, we see that the correlation between biomass and yield is negative at the beginning. Let's see. Having a high biomass means low yield, but later, having a high biomass means high yield, a positive correlation. So we have correlations that change over time. And here at the bottom, we have with these traits that are more constant across environments. So for example, radiation use efficiency was regulated by QTLs that were always the same. Nevertheless, we see that the correlation between radiation use efficiency and yield, in spite of radiation use efficiency being constant over time, the correlation with yield changes. We have a positive correlation, then zero correlation. The same for water limit, especially in dry environments. The correlation was zero, and then it becomes positive. So because each trait is activated or deactivated depending on the environment and depending on time, we see that something that is constant can vary in its importance to yield. Yeah, and that's quite a bit hard to understand, but it's also very useful to see because it can also be useful to select. To say, well, actually, for example, in this type of environments that are very dry, I will need low values for LL. Why? Because it will lead, a low value will lead to a high yield. A higher value will lead to low yield. Whereas in this other type of environment, it doesn't matter so much. Yeah, so it can give you some insights on how the relevance of the traits changes across environments and over time. And that's related to phenotyping. How often, how precise do we need to measure? Well, basically, if you have low heritability traits, you need to measure more often. This is what is shown here. So the more you phenotype, the more frequent you phenotype, the higher the heritability of this trait dynamics. And at the end, why do we want to make all these things? Because we want to make everything together, put everything together and combine them in a multi-trait model. Basically here, for example, we are predicting yield from yield, the red line. We have a heritability of 0.3, 0.60 something, and 0.5, depending on the environment type. And for some environments, we gain a lot by including these additional traits like biomass into the prediction for yield. For example, if we predict here yield from yield plus biomass, we get a prediction accuracy of 0.6. That is a lot better than 0.30 something, for example. But unfortunately, not always working, like here, and not always working in reality, because here when I came back to Chile, we tried to apply and we are still working on this, so this is very much work in progress. But we were having 3 locations, different years, and we were monitoring the stay green trait, that is in wheat, and these traits will help us to detect those genotypes that are more drought resilient. Yeah. So we characterize them with drones twice per week, and we try to find those curves, applying exactly the techniques that I already explained. So we first correct for spatial effects of NTBI. Then we characterize the variation over time. This is work done by the PhD student Francisco Mejias. And we see that the heritability first is not constant, because at the end, for example, which all the genotypes are yellow, are senescent, then well, we cannot distinguish them as well as when they are starting to senesce just after flowering. Yeah. So what do we do here? Well, we apply LMM solver to this dynamic. Here I'm showing only a selection of genotypes because we have 196. And we see that when we extract those curves, we see that, for example, in Valdivia, in the south, that is more humid, we have that Genesis always has a higher NTBI, so it's always greener. Whereas in Cauquenes, that is a more dry environment, we have La Sana Iña, that is the most green one. So we have a lot of G/E on these curves, so a lot of G/E going over time. So then we were happy. We said, well, we have a lot of variation in NDVI. Let's check how much this variation in NDVI, represented by the area under the curve, so the sum of all these fitted values for each day, relates to yield. Because if they do, they can help us predicting yield. And then we were again happy because, well, that's the pattern we see for G/E for NDVI was similar to the G/E for yield. So I said, okay, let's put everything in a linear mixed model for genomic prediction using some markers, and we can predict how genotypes can behave. And then we were a bit less happy because the results were not as positive as we wanted, but well, we still need to work a little bit on which traits or which summaries we need to include, because again, as for the simulated model, we saw that in some environments, like Cauquenes, for example, we had a big improvement in prediction accuracy when we include different traits, but in others, almost nothing. And we were a bit disappointed. But this is why, well, that's science, that's life, and we are still working on this to understand what is causing that in some environments works and some others doesn't, and well, we need to find other traits that can also help us. Maybe we didn't pick the right trait, yeah? But that's a bit the challenge. But the idea here, this prediction accuracy shows this path on how to summarize traits over time, summarize G/E, and put them all together to predict the target trait. That's the idea. So, to wrap up, understanding the TPE is critical for obtaining predictions that accelerate genetic gain. Environment groups can be identified using trait correlations, explicit environmental information, and gradients of genotypic rankings, for example, using splines. Target trait prediction and selection can benefit from selecting the underlying traits with complementary alleles, looking at static traits or also at dynamic traits. Incorporating dynamic traits in prediction models can help increasing accuracy and contribute understanding the adaptation mechanisms that explain G/E. But then again, we need to understand in which environments work and which one doesn't, because it's always a bit environment dependent, and that's a bit the challenge to understand when it will work and when it won't. So to finalize, I would like to thank you for your attention, and I would like to invite you. We have one of the biggest conferences about drought, breeding for drought tolerance in Chile. It's called the Interdrought. It's really a honor that we can host it in Chile, because this is the 8th version, and it has been in other continents, but never in South America. So we are very happy to host it here. So I invite you to visit our page, and if you have nice abstracts to submit, we are still receiving abstracts, so it would be very nice to meet you in person in November in La Serena. We will also have a nice field trip at the end of the conference, so that's also a good opportunity to get to see these very special dry environments we have. And I would like to thank all the people that have worked with whom I have collaborated, so the funding agencies and the team of people that work with me from Universidad de Talca, INEA, and my own university. And now I'm open and happy to receive some questions.
Thank you, Daniela, for the great presentation. Can you hear me?
Yes.
Okay. So we're gonna start to the question and answer sessions. We have a question for you. It's from Rafael Tassinari. The question is, when we talk about interactions G by E versus G by E and M, it's reasonable to consider the M factor as a part of the environment.
That's a very good question, because for the plant it doesn't matter. The plant only wants to have nitrogen, light, water, etc. But for us it matters, because we have more control on the M part. So this is why we basically split it. So yes, I think it makes a lot of sense, because, well, yes, a very simple but very relevant thing like planting date has a big impact on adaptation. And even for the same weather, it will change a lot if you have early or late planting. So I think, but we have a lot of control on the planting date. This is why I think it's good as breeders and agronomists to know which are the factors we can manipulate and control, and which are more random. So I think, yes, it makes a lot of sense to split it that way.
Okay, thank you. We have another question from Daniela. Predictive abilities in genomic selection rarely exceed the 5%, the 50%, and same for GM models. For instance, in G2F 2024, the best model reaches 35%. Do you think that we'll ever get over the 70-80% for new environments with G by E modeling?
Yeah, I share the feeling. That is, we wish, yeah, we try to do all these things. We invest a lot on drones, we invest a lot on markers, and it seems we will never go beyond. Yeah, I think we need to distinguish two things. One is, if we know the environment, so we know the rainfall, the temperature, etc., every condition, how good are we predicting for that particular environment that is known? And the other thing is the uncertainty we have in that we don't know how the environment is going to be. And that is even more difficult to assess and to predict. So I think, yeah, we try to make the models as good as possible, but the fact that we cannot be certain how the environment is going to be, I think we will always be a bit stuck with this lack of predictability. Yeah.
Yeah, I share the feeling too. Yeah. Well, thank you so much again for taking the time, sharing your experience with us. Uh, we now are going to have a lunch break, so feel free to join us at the rest of the evening.
Thanks for the invitation. And well, I wish you, I will try to join in the afternoon, but given that we will not interact anymore, I will wish you a very nice day. Thanks for the
Thank
invitation.
you. Thank you so much. Before we close this morning session, I'd like to remind you guys that we'll be back at 2 p.m. with two more great presentations. The live stream link has already been sent to all registered participants, but is also on the description of this video. Our team will also keep posting on social media, so please follow us on Instagram, LAMP-UFG and PPGGMP-UFG to stay updated. Thank you all for joining us this morning, and we will see you again at 2 p.m. sharp. Thank you.